This video, we're looking at 3.4 plasma membrane and membrane transport. This is part of Unit 3, the cell. Looking at plasma membrane. Let's do this. So we got cell and plasma membrane. The reason I put both of them up there is because I will say them interchangeably. Plasma membrane is probably the more accepted term nowadays, but I will say cell membrane because it's just the way I was trained, and that's just how it works. But they're essentially the same thing as far as we're concerned. The plasma membrane is composed of what is called a phospholipid bilayer. Where do we get that from? This is a phospholipid. Phospholipids are specialized kinds of lipids, and they have a hydrophilic head. Remember, hydrophilic means that it's polar. It's because it's got a phosphate on it. Phosphates are negative, so it's polar. And it's got a hydrophobic tail, which means that it doesn't like water. It is non-polar. And so when you put them together, they form this kind of bilayer in which the hydrophobic ends face one another and the hydrophobic heads face out toward the water that is inside and outside of the cell. And so it's kind of a protective layer that allows only certain things in and certain things out. There's a word for this. It's called semipermeable. So the plasma membrane is called semipermeable, or sometimes you'll see it called selectively permeable. I use semipermeable in my classroom, and it's the idea that only certain materials are able to pass in and out of the membrane. You can see here on this particular picture, water, represented by the blue dots, is able to pass to and from the membrane, but the big red sucrose molecules are not able to pass through because they're too big, or they've got a charge, or something causes molecules not to be able to pass through. You don't have to know all that for this class. You just have to know that with a selectively or semi-permeable membrane, not everything is able to get through the membrane. Speaking of that, let's talk a little bit about concentrations and solutions. We've talked about solutions before. These are mixtures of they're mixtures in which the solute is evenly distributed in the solvent, and with concentration has to do with the amount of solute per volume of the liquid. And so we're going to be talking a lot about concentration gradients here, so the understanding of concentration is pretty important. A lot of times it's going to be represented by these dots and things. If this was a chemistry class, it'd be represented by numbers. It's not, so we're just going to look at dots. We're going to go back the other way. So let's look at concentration gradients. So a gradient is like when you have a high area and a low area, right? Like a roof or a hill or something like that. Well, you can have a concentration gradient, and that's where one side of a membrane has a high concentration and the other side has a low concentration. It could just be like areas, like in a jar. I'm going to show you a picture in a minute. It's got like a high concentrated area in the middle of the jar, and it's eventually going to go out into the whole jar until that concentration gradient is not existent anymore. So a gradient is when you have a high concentration on one side and a low concentration on the other side. In general, materials will move from an area of high concentration to an area of low concentration, kind of like if you were on the top of a hill on your bicycle. You would ride down that bicycle without any input of energy at all, right? It would just kind of go down. Well, this has a name. It's called passive transport. Passive transport is the movement of materials without using energy. Typically, this is going from a high concentration to a low concentration. There are two kinds of passive transport that we're going to look at versus diffusion. So diffusion is this idea, again, moving from particles moving from an area of high concentration to an area of low concentration. Here you can see they've put a dropper of some sort of like food coloring or something in a beaker, and there's a high concentration inside that drop, and then as time passes, that food coloring will disperse out into the rest of the liquid, causing those concentrations to be equal throughout until they are equal throughout, and there's a word for that. It's called equilibrium. And so particles will move from an area of high concentration to an area of low concentration until those particles meet equilibrium or reach equilibrium. There's a particular kind of diffusion that we need to talk about. It's called osmosis. This is the diffusion of water. So we're still dealing with high concentration, low concentration, but in this instance, we're dealing with those concentrations being of water. Water also has a concentration. So if you look at the picture, the beaker on the beaker number 1 there has a higher concentration of water on the left side of the beaker, right? Because there's a lower concentration of purple dots. The right side has a higher concentration of purple, so it has less concentration of water. Well, purple can't move through the semi-permeable membrane that has been drawn there. And so what happens is water will actually go from an area of high concentration to an area of low concentration until it equals out, until it reaches equilibrium. And so water tends to move from an area of high concentration to an area of low concentration as well. However, when water moves, it's not typically referring to the amount of water. Rather, it refers to the amount of solute in an area. So let's look at that. These words, hypertonic, isotonic, and hypotonic, have to do with the amount of solute in an area. Hyper means more. Hypo means less. Another important thing that you have to understand about these words, they're comparative. And so just like I would not say he is taller than, and then just in that sentence, you're expecting me to say, who is he taller than, right? And so when you use the word hypertonic, you are referring to something else. It's a comparative kind of word. And so you would say the inside of the cell is hypertonic to the outside of the cell, or the inside of the cell is hypotonic to the outside of the cell. Hypertonic means more solute. And so if it's more solute, it's less water. Hypotonic is less solute or more water. And so water will tend to move from an area that is hypotonic to an area that is hypertonic, or from an area that is more water to an area that is less water. And when solute concentrations are equal on both sides, we call that isotonic. And so those two words have to do with the amount of solute, but it still has to do with the movement of water in relation to that solute. It's really important. All right, another kind of passive transport is called facilitated diffusion. Think of the word facilitate. To facilitate something is to help it, right? And so facilitated diffusion has to do with the movement of materials across a cell membrane using a protein channel. For some reason, those molecules can't get across the cell membrane. Maybe they're polar, maybe they're big, who knows, whatever's going on. They need help getting across the cell membrane. And so still diffusion, still high to low, but we're using a protein channel in order to get across the membrane. And then there's active transport. If passive transport uses no energy, active transport uses energy. If passive transport goes from high to low, active goes from low to high. It's just like when you're shoving items into a closet that's already full. It requires energy. You have to use energy to shut that closet. But what have you created? You've created a concentration gradient, right? There's a high degree of pressure inside the closet, low outside. If you just touch the doorknob, it's all going to spring loose. And so active transport is oftentimes used by cells in order to create gradients, and those gradients are used to do cellular work. We're going to talk about this when we get to photosynthesis and cellular respiration. So active transport requires energy, and it is movement of materials from a low concentration to a high concentration. A couple of examples of active transport that are specialized are endocytosis and exocytosis. And this is the bringing materials into the cell or out of the cell, large materials. So endo is bringing large materials or several materials, as you can see, like lots of stars going into the cell here. And it's the creation of a vesicle, right? It creates this little bubble, as you can see. It takes it into like a little temporary storage bag. Remember we talked about that with the endomembrane system, right? Except for we talked about exocytosis with the endomembrane system because it goes from the Golgi, gets into a vesicle, and it gets pushed out of the cell, and that's kind of where we left it. Well, now you know that process is called exocytosis. And so these are both forms of active transport because they require energy. And you can also see that they're using the cell membrane in order to do this. They're like pinching off a piece of the cell membrane or adding to the cell membrane. So big idea here, endo, large materials into the cell, exo, large materials out of the cell.